Today we will continue our discussion about this GPI-APs that are being regulated or deacylated by Calnexin. So in the previous video we have discussed in brief how this Calnexin helps in maturation and deacylation of GPI-APs. How they have come to this conclusion, and that is discussed in the paper. So we will cover each figures in detail one by one. So today we will cover the first figure, which talks about the conformation of the expression of this mature GPI-APs on the plasma membrane of wild type cells and also on the Calnexin and calreticulum dual knockout. So what is the strategy that they have used is that if the mature GPI-APs are expressed on the cell surface, then they can be cleaved by this PIP-LC. However, the immature GPI-APs, when they are expressed on the cell surface, they are not able to be cleaved by PIP-LC, because the insoluble makes a bond at different position, second position, whereas in this case, in the wild type case or in the mature GPI-AP case, the insolubles make a bond at the first position. So first, for the check this for the wild type cells, and for the wild type cells without any PI-PLC treatment, they can see the more signals for this CD59, CD55 and CD109. However, after this PI-PLC treatment, they can see the reduction in these signals of CD59, CD55 and CD109, and the background signals is indicated by dashed line. So this is the how the wild type reacts. So this indicates that wild type has the mature PI-PLCs, and this GPI-APs expressed this kind of proteins. So what they have used the researchers have used the antibodies against this proteins that are expressed on the cell surface, and they can see the change in the expression in the flow cytometer. And for the dual knockout, when they check it, that for CD59 they can see some reduction in signal, so it indicates that some of the PIP-LCs are mature, but some of them are immature, not PIP-LC. Some of the GPI-APs are mature and some of them are not mature. And similarly for the case for this CD55 and the CD109. So after this flow cytometry analysis, they did the quantitative analysis of the fluorescence intensity of this remaining GPI-APs. So remaining GPI-APs means that how they have considered, or the definition that they have considered for the GPI-APs, remaining GPI-APs, is that what are the signal that is left divided by the signals without PI-PLC treatment. So for the CD59 and for the wild type, they have considered the signal as the reference 1, whereas for this dual knockout, they have considered the intensity based on this wild type reference as 1. And what they have find out is that the remaining GPI-APs in this CD59 and CD55 and CD109 are statistically significant or statistically higher compared to the wild type. So this confirms that there are some kind of immature GPI-APs that are expressed on the plasma membrane of the dual knockout. And after this, they also validated the other GPI-APs that are also possible, that are also found on the plasma membranes, that GPI-APs that are expressed on the plasma membrane, such as CD48, CD14, ART3, ART4, LY6, LY6, G6C, CD52, and in all of them they can see that there is increase in the remaining GPI-APs, and this increase is statistically significant compared to the wild type. So this indicates there is some kind of immature GPI-APs that are being expressed on the plasma membrane of the dual knockout. So from this figure, they validated their first hypothesis that Calnexin and calreticulum, in the Calnexin and calreticulum dual knockout cells, there is a quality control loss occurs that leads to the expression of this immature GPI-APs on the cell surface. So I hope you understood this, how this experiment is conducted, what is the hypothesis, and what are their findings. And if you need to understand more about this experiment in details, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.